Jackie. Now I'm Jo Preston. I run marketing and fundraising here at Bionics Institute. And as you can see, I work with some really wonderful people doing amazing things. And they're about to show you in real life the amazing work they do. So go ahead.
Alright, so I am going to stimulate the motor cortex of Oscar's brain. So as we know, or we might not know, the left side of the brain controls the right side of the body. So I am going to stimulate his left motor cortex. He's going to get a jump in his right hand. So if I get the right spot on the brain, we'll see.
You'll hear a clicking sound for when it fires.
Just give me a second.
Pre-drew the spot.
Don't give away my secrets. I can't find it anywhere.
Oh, that's fine. Well, we'll do it live. This is good.
Aha. Okay. Yeah. So that click you sound. Sorry. Try again. So that click you sound is actually the sound of the magnetic pulse passing through into Oscar's brain. And so every time we get that, yep,
Oh,
there we go. So every time we hit the right spot, every time we hit that neuron in the brain that's controlling the hand, we'll get a twitch.
This isn't me just moving my hand
No.
on purpose, by the way.
Do you want to do an active one?
Oh, yeah.
Yeah, then it might be bigger. So you might be able to see that. Yeah, so it's a bit of a bigger twitch there. And so that's essentially TMS. So that we were doing those pulses there about once every 2 seconds, 3 seconds. So in the trial, we're applying pulses around between 50 and 60 times every second. So much higher. So it's quite loud. And you often can't actually hear the individual pulses because they all sort of run together. So that's a very mini version of what we do with the TMS.
And I couldn't really feel much there. Just felt like a slight tapping on my head. Yeah.
Fantastic, thanks. Stay right there.
Yep.
And it's now time for questions. So I think you had a question to start with. Jackie, come on,
how
John.
much voltage is I don't quite understand when you stimulate that machine.
Yeah.
Is
So yeah, you go.
Well, I was going to say that I'm not sure of the exact voltage required in order to actually generate the magnetic pulse, but essentially it relies upon the fact that when you create an electrical current, it creates a perpendicular magnetic field. So the electricity runs around a coil inside here, which creates a magnetic field here that comes out. So the electricity running through here isn't what's going into my head. There's no electricity going in there, but it is generating a magnetic field that then causes the neurons to fire. Kind of how, like, if you ever held, a magnet up to a TV, you could interfere with what was on the TV from the magnet, even though you weren't actually passing it directly in there itself.
So the strength of the magnetic pulse, so that's measured in Teslas, and it's around 2.2 Tesla. So to give you some context about what that is,
that Tesla?
Tesla, yeah. A lot of the MRIs that are available in hospitals were around sort of 3.5 Tesla. So it's a pretty strong magnetic field that we use. It does dissipate very quickly. So it's only sort of strong at the point at which you stimulate, and then it weakens very, very quickly.
Any other questions?
Yeah, I'd ask a question from a patient's point of view. If you're actually stimulating at 30 hertz or whatever, and it's clicking each time, that's a lot of noise. Why does it need to be noisy? Because that would be very distracting.
Yeah, so it's just the noise that the magnetic pulses, it's what it makes as it discharges. The device discharges. There's not much we can do about that. What we do is every single patient who has TMS has to wear very good earplugs. So we put earplugs in to control, to make sure there's no damage to hearing. But it's a very good point, it can get quite loud.
The other thing is the way we deliver the stimulation protocol, the entire stimulation for a region of the brain takes about 3.5 minutes. So it is relatively short in terms of them sitting there.
Now I'd be more interested in what the technology is to produce the pulse. It's fairly high currents, obviously.
Yeah.
Yeah.
That's why they don't have portable devices yet.
Yes.
Mike?
A question for Oscar. The brain's a fairly large organ and multi-layered, so obviously a lot of what you're doing is at the surface. How deep within the cortex do you actually get?
With the TMS machine?
With the overall research and analysis.
So with fMRI, you can get down to the brain stem. You can be getting pictures of the entire brain. There's a very good question though, because that's another one of the differences I hadn't fully gone through. So for FNIRS, it is primarily the surface of the brain, the outer layer of the brain. Now, I guess somewhat fortunately, the outer layer of the brain is where a lot of our cognitive abilities are sort of stored there. And then for EEG, there are techniques to essentially work backwards from recording on the surface of the head to trying to reconstruct a picture of the whole brain there. Now, one of the things is even when you're looking at just the surface of the brain, we know which parts of the brain are sort of connected to one another from the fMRI. So by using that information, there are sort of advanced computational ways to try and, looking at something like FNIRS, try and work backwards and try and get a really holistic picture, which really sort of ties into that purpose of why multiple techniques might actually be really important, because otherwise you've only got part of the picture there.
Thank you.
Any other questions? Yes.
So I do understand that we don't know the reason why Alzheimer happens, but you said that there is a protein which gets accumulated. So is there any research going on whether it is due to the food we eat or the habits or genetics? What is is any research going in that area where it can be identified?
It's a really good question. And so there is a lot of research looking at risk factors for Alzheimer's and looking at factors which might contribute to amyloid accumulating and leading to Alzheimer's in some people. So when we talk about, it might be worth just sort of touching on some of those modifiable risk factors that I kind of alluded to earlier. So age is the strongest risk factor for Alzheimer's, but of course that's not modifiable. That happens no matter what we do, no matter how we try and change it. And so as we get older, you can, if you have a build-up of amyloid, sometimes that can impact the connectivity in the brain. And as we get older, that relationship seems to increase. There are things we can do to try and keep the brain as healthy as possible, to keep that connectivity strong, perhaps even help sort of ward off the negative effects of amyloid is the hope. And so those are things like eating very well, having a very good healthy diet. So Mediterranean diet is often pointed to as a really positive one. Exercise, cardiovascular exercise, keeping up social engagement, so social connections, working your brain, doing things, whatever it is that's new to you, learning new things. From a medical point of view, making sure any cardiovascular risk factors or any cardiovascular disease is taken care of, and good hearing. So there's sort of some of the constellation of things that we can do, and we can do all of those things in our middle age to try and improve things.
And one more question. So in the presentation, it was also mentioned around the oxygen in the brain.
Yep.
So if people who snooze and get less oxygen when they're sleeping, can this lead to accumulation of these proteins?
So there is a lot of research on looking at things like sleep apnea is what I think you might be referring to, the snoring and So there is certainly a lot of research looking into that. I'm not sure about what the most recent findings are with respect to sleep apnea and dementia. Yeah.
I was going to say, sleep apnea untreated is actually one of the biggest predictors of a subtype of dementia called vascular dementia, because what you get is the brain isn't getting enough oxygen, and the brain uses a disproportionate amount of your body's oxygen. So, it's about 350 grams, but it uses about a quarter of your body's energy. And so if you don't have any oxygen to it, even for a short period of time, you can start getting these brain cells start dying. So something like sleep apnea it's a really big factor, because otherwise people are getting 30, 40 years of being slightly oxygen deprived every single night. And then also things with like sleep apnea, you get adrenaline every time they stop breathing and have to wake up again, and this has a lot of sort of negative impacts. So, I mean, overall, the sort of general theme seems to be things that are good for your health are good for your brain. And so anything that can be good for your own well-being is probably also going to have a beneficial effect and reduce your likelihood of developing Alzheimer's.
Absolutely.
Could you just expand, you mentioned hearing, and this is the first time, could you?
Yep. So hearing loss, there's been new research out actually relatively recently showing that hearing loss can be a significant risk factor for Alzheimer's. So one thing I just want to clarify, we're not talking about things that will lead to Alzheimer's. We're talking about risk and relative risk. So things that can increase our risk or decrease our risk. So they don't mean we will get it and they don't mean we won't get it. They're just things that we can do to minimise our risk. So the hearing loss in particular really is around that if you have hearing difficulties, making sure that you address those. So that if you need hearing aids, making sure you wear them and they're fitted well and they're working well. So it's not necessarily, it doesn't seem to necessarily be about the fact that you have hearing loss. It's about the effect of not being able to hear consistently. So it's just making sure those things are taken care of if they can be.
In the comment.
Yeah, thank you, Joe. Great presentation. I was just wondering, because there's something that's missing with me. The problem in getting more dementia and Alzheimer's is due to having more amyloid in the brain. This is the answer, but how does it answer that, the TMS?
So to clarify, we know that amyloid plays a very, very important role in Alzheimer's. So I don't think we're there yet to say it's completely causative, but we know that it is an important thing that's happening in the brain. So you're right in that TMS won't necessarily impact amyloid in the brain, but what we're hoping it will do is essentially strengthen the connectivity in the brain, strengthen the brain communication, so that we can get an improvement in memory for people who are suffering memory loss at the moment. Hopefully, potentially even delay progression by essentially driving in a sort of external way healthy brain function. So essentially what we're trying to do is all of the things we spoke about that can really improve the health of your brain, we're trying to do that sort of externally and really drive that. There is some research that shows that really strong functional connectivity could have a protective effect against the ongoing impacts of amyloid. So that if you have really strong functional connectivity, strong communication in your brain might be less susceptible to the negative effects of amyloid, essentially.
One thing I would just add to that is some of those drugs that might remove amyloid, some of the very, very big studies have found that they successfully removed amyloid, but people didn't actually get better. So their memory didn't improve. And so it's definitely linked to it, but it's not the full answer to the question, because you also get cases where someone, they might do a sort of testing levels of amyloid, and they find after someone has passed away that they had extremely high amyloid, but they actually had no symptoms of Alzheimer's. And so it's sort of, it's definitely involved, but the exact role of it, I think, is still something they're looking into.
Thank you.
You want to ask that?
Yes, I've been a volunteer with the ABLE study now for over 10 years, and they have accumulated an enormous amount of information, which is now, I believe, in a university in America, which is available to every researcher in the world for nothing if they're working on Alzheimer's. It seems like you're also looking at biomarkers, which obviously the ABLE study looks at.
Yes.
Do you actually use that research that they have accumulated? Otherwise, you're really, repeating something which is already available.
Yes.
And that's been the whole idea of being part of the research, was to make all of that information available to scientists like yourself.
Yeah, it's a really, really good point. So ABLE's been a really important project and study that has helped us understand brain function in Alzheimer's more than we have in the past, and we've certainly used those learnings in the development of this trial, which is really important. So ABLE is also associated with ADNET, so the Alzheimer's Dementia Network. And so we are also linked in with ADNET. So we are working together. We're not sort of working in silos. The data we're collecting here is not exactly the same as they're collecting as part of ABLE. Some of it is similar, some of it isn't. And so while, yes, it's very important that we work together, sometimes there is the need to, particularly if you're trying something that's a bit different and a bit unique, there is the need to collect the data directly as well. But yeah, absolutely working together.
I was just going to say on that, it's often building upon what they have found by changing something slightly to get a new piece of information. So for instance, it's not just having data like fMRI from 100 people and then EEG from another 100 people. It's trying to sort of be from the same people so that you can sort of stitch all that together into a comprehensive picture. But as you were sort of saying, that information is the foundation from which we can say, okay, this is where we need to look, or this is how we should sort of try and record.
Lovely. Thank you. Oh, sorry.
Oh, can I? Oh, sorry. Yeah. I have a lot of questions, but one that I want to ask first of all is about this placebo group and what they are getting as a placebo group, and if it's the same looking sort of treatment, does the clinician know they are delivering the full treatment?
So these are not the exact coils that we use in the treatment, they're a little bit different, but essentially the placebo coil and the active coil, they're identical, so they look exactly the same. The difference being that there is a specific shielding on the placebo coil which prevents the magnetic stimulation. You get the sound, and there's also a way in which they replicate the feeling of the stimulation on the head, but they're not actually getting the magnetic stimulation. The treater, so the person administering the treatment, always knows what group the person is in when they're administering the treatment, but the treaters have no role in assessing whether anything has changed. So their role is to just provide the treatment to the patients.
So they don't do any follow-up at all. And so that brought me to my second question, functional assessment of people. You've talked a lot about using these specific treatments, pre and post. What other functional assessment do you use?
So specifically, we use a scale called the ADAS, Activities of Daily, the Alzheimer's Activities of Daily Living, which is a comprehensive interview that goes through all of the different areas of community functioning and personal functioning and family. We do that with someone's carer as well as with themselves so that we get complementary information. We also do a quality of life scale, and we do very comprehensive sort of memory testing and things as well.
But each of those things can be influenced by the recipient's perspective.
Yes.
So they're not a functional test like please walk that line from here to there or whatever.
We refer to them as behaviourally, like functional behavioural tests, because that's what they are. So the tests, the neurobiological assessments we use, they're the ones where it's just based on what is happening in the brain and the blood tests. And then we also do need to assess how people feel they have changed, because that's one of the key important things that we're interested in.
And there are some more objective ones as well. So for instance, asking someone to write a letter, address it properly in a way that would be...
That's the sort of thing I'm asking.
And so that's included, but then also what's really important is the information we get from their close other as well, because sometimes there may be subtle differences that I'm noticing they are losing their keys less, or they're better, more confident in doing this. So
Just one last question. Thank you.
What will success look like for your trial?
Sorry, I didn't quite hear that
What
one.
will success look like for your trial?
So it's a really good question. So what would success look like for the trial? So we are hoping for a significant improvement in cognition that lasts to our follow-ups. But we are also powered, or we've also designed the trial to look at a lack of progression in the active group as well. Either one of which would be a positive outcome, the way that we're looking at it. And one of the key things that we're really keen on in this trial is for the effects to be as broad as possible across the group. So that's one of the reasons we're personalising, to try and make sure that we see effects, positive effects, if they are there, in as many of the participants as possible, rather than just a group level effect. We want to look at how consistent that is across people as well.
Can I just follow up with the, I understand that there's neuronal loss in Alzheimer's, so when you're doing your MRI, are you looking for progression in neuronal loss? Like, would you expect that this stimulation would stop the neuronal loss?
Look, it's a very remote possibility that it could. It's not something that's our sort of first line hypothesis, because it is a very remote possibility, but TMS has been shown to lead to neurogenesis, to lead to brain growth. Whether that occurs in Alzheimer's is a very big unknown question, because people haven't really looked at it in detail. But yeah, we'll have the data, because people will be getting structural scans, so we absolutely will be able to look at that as well.
Wonderful. Thank you so much. What a fantastic presentation. Now, like Kate said, if you know anyone who might like to take part in the trial, please contact us. It would be, wonderful if we could find more people to take part in the trial, and also they might get some benefit from it as well in the future. And I'd like to say a big thank you to those of you here who have been donating to or giving to the research. We really, really appreciate your support, and it just wouldn't happen without you, so thank you. And to those of you who are new to the research and have come to find out what it's all about, if you would like to support it, then please contact me or one of my team, and we'd love to talk some more. And now the only thing left is to say, join us for some tea and cake, because you've been looking at it the whole time. So thank you.